Hi everyone, in this last unit of the semester we'll be focusing on the development of ectodermal tissues, more specifically on the development of the nervous system. Although we're going to be looking at the molecular aspects that control the development of the nervous system, we'll also see how this system is deeply affected both positively and negatively by changes in the environment of the embryo. As we'll see over the next couple of weeks, and particularly in the last lecture of the semester, the events that happen during development can have lifelong consequences, even on the ways we behave and experience the world. 3 of the 4 chapters we'll be covering in this unit relate to the development of the nervous system. In this lecture, we'll learn about how the neural tube forms from a sheet of epithelium, and we'll also look at the patterning of the neural tube, or how cells are specified in the neural tube along the different axes. In the next chapter, we'll focus on the specification of the different cell types of the brain and also on the growth of the brain from the neural tube. Finally, in chapter 15, we'll explore the development of neural crest lineages, which include the cells that form the peripheral nervous system. The development of the central nervous system begins with the formation of a simple tube, and that tube forms from a sheet of epithelium. Morphogen gradients then pattern the tube, giving different regions a distinct identity and function. Recall that the neural tube is derived from the ectoderm, which is a germ layer that's located at the surface of the gastrula. The vertebrate neural tube is derived from a dorsal region of the ectoderm that's known as the neural plate. That neural plate will form a tube, and from this tube, the central nervous system, consisting primarily of the brain and the spinal cord, will form. The ectoderm will also give rise to the epidermis and its associated structures like the lens of the eye, hair, and nails. Between the epidermis and the neural tube, a population of cells that's known as the neural crest will arise and migrate to different locations of the body. And that population of cells gives rise to different tissues, including bones of the face, the peripheral nervous system, and pigment cells, among others. And the process that leads to the separation of these 3 ectodermal derivatives is known as neurulation, and it begins after gastrulation. So an embryo that's undergoing this process is known as a neurula. These diagrams show the process of neurulation in an amphibian embryo. The pictures at the top here are looking down at the embryo. This is the anterior and this is the posterior of the embryo, and we're looking here at the dorsal side. The drawings at the bottom are cross sections through the embryo, where the dorsal side of the embryo faces upward and the ventral side is on the bottom. The process of neurulation involves the formation of the neural plate and its folding into a tube and separation of the neural crest, which is not shown in these images. But what is it that controls the specification of the ectoderm into these 3 different tissues? Well, we've actually already encountered the answer to that question, because in previous lectures, when we looked at early amphibian development and early mammalian development, we saw how BMPs have to be inhibited in order for neural tissues to form. So the specification of the ectoderm is regulated by the levels of BMP. The portion of the ectoderm that is exposed to high levels of BMP will develop into the epidermis. The portion that's exposed to intermediate levels will develop into the neural crest, and the region of the ectoderm that's exposed to the lowest levels of BMP will form the neural plate. As we'll see, even though BMP inhibition is important to allow the formation of the neural tissue, some BMP is needed to promote the proper folding of the neural tube. These low levels of BMP are indirectly regulated by SOX transcription factors. So these transcription factors promote the development of the central nervous system, both by inhibiting the expression and the signaling of BMPs, although this happens indirectly, and also by activating genes that are needed to specify the neural plate. Different mechanisms of neurulation are utilized along the anterior-posterior, which is also known as the rostral-caudal axis of the body. And there are also variations that are specific to certain organisms. But in general, the more anterior portion of the neural tube is going to form through a process that's known as primary neurulation. So this involves the proliferation and the folding of the neural plate into a hollow tube that is covered by the overlying epidermal ectoderm. And this region that forms by primary neurulation is going to give rise to the brain and the majority of the spinal cord. The more posterior or caudal regions of the neural tube form through secondary neurulation, and this process involves the coalescing of mesenchymal cells to form a rod, and then this rod forms a cavity. A portion of the neural tube between the region that's forming via primary neurulation and the region that's forming via secondary neurulation forms through a process that's known as junctional neurulation. And this region is known as a transition zone, and the size of it varies depending on the organism. Junctional neurulation, as you can see from the figures, involves aspects of both primary and secondary neurulation. As you can see, there's both invagination happening as well as the coming together of mesenchymal cells.